Good afternoon, everybody. Welcome to today's Living Environment lesson. Today we're going to talk a little bit about biotechnology, some of the techniques and practices that we have around the world and in the scientific community to take the knowledge that we've been talking about, right? The idea that DNA contains all of these genes and all of this information, and how we've kind of used it for sometimes for our own gain, sometimes for the benefit of the planet, sometimes for the benefit of endangered species, etc. Right. Biotechnology is exactly what it sounds like. It's taking biology and technology and combining them together, right? Using technology, using our knowledge of biology to do certain things, right? Whether that's making bread and using, yeast making carbon dioxide to make bread rise, whether that's making cheese, whether that's, making a corgi, right? Each of these things is a biotechnological advancement that we've made. Some of them really simple, like selective breeding, some of them much more difficult, like cloning and genetic engineering. The first one we're going to talk a little bit about selective breeding. Selective breeding is when we as humans, as the human race, take certain organisms and breed them together to get the desirable traits from both organisms into one subsequent organism. Right? We use this to grow certain fruits the right way, not the right way, but the way that we like them, right? Sweet apples with juicy apples to then get both of those traits, so we have apples that are both juicy and sweet, right? We use selective breeding to make certain foods allergy free, right? We use selective breeding to get different types of dogs and different types of cats. We use selective breeding to get good racehorses and to get good greyhounds and to get good corn and to get good apples, right? To make these organisms, to take these species and kind of push them, nudge them in the direction that we want them to go. Part of the downside for this is that a lot of selective breeding can often lead to inbreeding, where we're breeding, the same two organisms repeatedly to get their traits, and then breeding the children together to keep those traits and the children together to keep those traits. And as we do that, we have the DNA is not able to change at all. So instead of taking a new organism to then breed into a pool to make new organisms, we will have the same genetics, the same DNA passed down for ages and ages, and as it passes down, there's no variance, there's no variation. It also means that any undesirable traits that the organisms have get passed down, and there's no way to take those out and to breed those out if we're just looking for the same positive traits and not focusing on negative traits, right? Show dogs with bad hips are an example. Racehorses with fragile legs are an example. You know that certain breeds of dogs have kidney problems. Certain breeds of dogs are more prone to liver problems or to problems with their internal organs because of the way that they have been inbred throughout time. Genetic engineering, the next step in, biotechnology as a whole, takes DNA from one species and actually puts it into a different species to kind of create these genetically modified organisms, which we'll talk a little bit more about later. Right? But the idea that we can take DNA from fish to put it into a strawberry, right? We can take the cold resistance from fish that live in the Arctic and put that into strawberries to keep them from freezing when it gets colder. We can take, and the big one that we do here, we can take human insulin DNA, right? The DNA that says this is how you make insulin, and put it into a bacteria, which we can then grow and grow and grow into these large populations of bacteria, and harvest the insulin that we have. Basically, we've taught them how to make insulin. And so we can harvest human insulin from bacteria to then give to patients with diabetes. Right? The act of doing this and taking the DNA from one species and putting it into another is a really difficult process, and it took a long time for us to figure out how to do it. But basically, we are using enzymes to cut, copy, and paste, like a computer, DNA from one thing into another. Right? And when we do that cut, copy, and paste, we are going to take a bacteria and human or other organism, and using enzymes we will cut the pieces of DNA that we want gone, right? We'll remove a piece of DNA from the bacteria. We will remove a piece of DNA from the target, and we'll then paste them together. So the idea is that this isolated gene could be the gene that makes insulin. We've taken it out of a human and put it into a bacteria, and now when this bacteria reads its DNA, when we put that bacteria into triggering conditions, it will create the protein that we have shown it how to make. Right? But enzyme control and being able to tell an enzyme, hey, cut here, cut there, paste here, paste there, allows us to take DNA out of one thing and put it into another, and then create an organism that can do something unique, something special that we have designed it to do. Right, and this is the foundation of genetically modified organisms. Right? When we talk about GMOs today, we're talking more about, cutting and pasting certain genes to make a different new organism that does a new thing that we want it to do. But GMO is not necessarily limited to just that. Wouldn't this be dangerous? We try our best. The short answer is yes, right? This could be very dangerous. This could very negatively affect the target organism, right? We could end up with a bacteria that just isn't viable, right? By putting this new gene into the bacteria, we could have a bacteria that just dies, right? But with practice and with scientific experimentation, we've learned how to do this a little bit safer, especially for things like bacteria, which are single-celled organisms and a little bit more simple in terms of their biology. When it comes to larger organisms, it is a little bit trickier, and it is a lot more difficult to make a viable to have a viable DNA transplant. That's the phrase I'm looking for. Not yet, sorry. When we want to check then for viability. We try our best to do this on a small scale. We try with bacteria, which we can easily grow lots and lots of bacteria. We're not at any risk of creating something that can't then be stopped. Right? But a lot of the times the issue is that the new organism, the product organism, is just not viable. It just won't survive. Another piece of biotechnology that we use is DNA hybridization. This is a technique that lets us compare two different strands of DNA. It lets us see how similar they are. And the way that this ends up being useful is for a lot of CSI and forensic tests. If we want to do a test to see how similar two organisms are to each other, we take their DNA and we see if it matches up, if it lines up with the other. Right? We look for similarities in the DNA, and the higher the percentage of similarities, the more likely the two strands of DNA come from related organisms or the same organism. Right? And the way this works, I will do some example here. Right, we take two pieces of DNA. Right, we take two different sources of DNA. Right, so this red is from source 1, and this purple is from source 2. We take them apart and we mix them together. Let me see here. I would like this to do that and this and that. Okay. Sorry, I just need to think through that example for a second. So when I take these apart and I let them mix together, what we're looking for is spots where the base pairs are going to line up again. And so if I mix together this strand and this strand to see how similar they are, we're going to have A, T, C, C, G, G compared to C, G, 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 T, T. When we look at the way these are going to line up, they're not very similar. And we can tell that because the only base pairs that are going to connect are going to be this CG and this CG. Right? There's no connection between the A and the C. No connection between the T and the G. No connection between these G's and these T's. So if I want to quantify that, right, 1, 2, 3, 4, 5, 6, it's 2 out of 6 that are similar, which is 1/3, which is 30% similar. Right? Not a great lineup. And remember that DNA is hundreds upon hundreds, thousands upon thousands of these base pairs. So saying that 2 out of, a couple thousand line up is not good. For this sample, that's 30%. That's pretty good. But remember, this would be much longer in both directions. And we would see the lining up that happens there. So now, when we talk our way through genetically modified organisms, all that means, and this is a really big phrase that people like to use, or they like to throw around like, oh, GMOs, they're so bad, GMOs, they're so dangerous, GMOs this, GMOs that. A genetically modified organism is any organism that has had their DNA modified for a purpose. Right? That means most cats, most dogs, most horses are genetically modified organisms. They were selectively bred to be the way they are. They are GMOs. When we talk about GMOs in terms of crops, we're talking about any crop. When we talked about take the sweet apple, you take the juicy apple, and you breed those strands, those apple types together to try to get a sweet and a juicy apple, that is a GMO. Right? You have used the DNA of that organism you've tried to change the DNA of that organism. Now, that also applies to that strawberry I mentioned before, where we take the DNA from an Arctic fish and we put it into a strawberry to try to make a strawberry that is resistant to the cold. That also applies to taking certain insect repellent proteins and allowing them to exist inside of, crops like corn and wheat to make them naturally resistant to insects and pests. Right? Or making plants that have a higher heat tolerance. Or plants that can survive in drier areas or wetter areas. Right? And all those things are great. They're benefits. They're helping, not spray chemicals all over our fields. But there are also worries about having pests that then evolve around these GMOs, pests that evolve to become resistant to the thing that hurt them before. Right? Pests that ignore something that they used to be repelled by, and now they just don't care. Right? We find these. Any change that we make needs a reaction from the environment. And there are also the people who have concerns about, is this something that I want to be eating? And at the end of the day, I personally don't think it matters. I've read some of the studies, and I don't think it makes that big a difference. But I think people make a lot of money selling organic food. And so they're going to keep fighting GMOs. Oh, science is dangerous. Oh, science is bad. I shrugged. You couldn't see it, but I shrugged. So long story short, biotechnology is not something new. It is not something that people are doing without care. It is something that is being done with careful observations and careful study, and it's also the way you have a Chihuahua and a Pomeranian. Biotechnology is not new concept. It's an old concept that we're applying new technology to. And it's the way that we can make a lot of medicines that we make. It's the way that we can do things like harvest insulin. Instead of harvesting it from human beings, we can harvest it from bacteria who are made with the specific purpose of making insulin so that people with diabetes can survive. And with that, I'm going to end this video. I hope you all have a fantastic day, and I'll see you next time.